This is Chapter 3, Part A on cells, the living units. So a cell is the basic structural and functional unit of life, which means all living things are composed of cells. So how well an organism is able to function is going to depend on the individual and the combined activities of all of its cells working together. So remember, kind of the recurring theme in anatomy and physiology is that structure and function are complementary, meaning that the function of cells is going to be dictated by their shape. Cell theory also states that cells can only arise from other pre-existing cells. So we don't have just a spontaneous generation of cells. So living things have to come from other living things. There are over 200 different types of various cells in the human body. So these cells differ in their size, their shape, and subcellular components. So all of these differences contribute to their differences in function. So we can have some cells, like epithelial cells and blood cells, that connect different body parts or form linings and transport gases. We have some cells, like muscle cells, that can move organs or move body parts. We have cells that store nutrients, like fat cells, cells that fight off diseases, like our immune system cells. We have nerve cells that can gather information and control other body functions. And then we have cells capable of reproduction, like the sperm and the egg. So despite all of this variation and diversity in the human cells, they all have the three same basic components. So all cells will have a plasma membrane, which is just the flexible outer boundary of the cell, kind of like the skin of the cell. The cytoplasm, which is the fluid inside the cell that's going to contain all of the organelles and the cell parts. And then the nucleus, kind of in the core of the cell, that's going to contain the DNA. So this is the major control center of the cell. Extracellular materials are things that are outside the cell. So extra meaning outside. So things like body fluids, interstitial tissue fluids, blood plasma, cerebrospinal fluid are all examples of extracellular body fluids, as well as cellular secretions like saliva and mucus, as well as the extracellular matrix that helps to hold our cells together. The plasma membrane is sometimes referred to as a fluid mosaic, meaning that it's made up of many different smaller pieces. So the plasma membrane is going to act as a selective barrier, separating our intracellular fluid and components from the extracellular fluid outside of the cell. So it's going to play an active role in regulating cell activity by controlling what's allowed in and out of the cell and when. So looking at the structure of the plasma membrane, so it's composed of membrane lipids. So these phospholipids we talked about in Chapter 2 are going to form this lipid bilayer. So we have a double layer of the phospholipids. And then throughout the membrane, there are proteins, membrane proteins kind of embedded that can float throughout this fluid membrane. So it has kind of like an oil-like consistency. There's also surface sugars on the surface of the cell called the glycocalyx or the sugar case. So these function in cellular identification. So these are kind of like ID markers on the surface of a cell, so other cells are able to recognize it. Some other membrane structures can help to hold cells together and form tissues through special types of cell junctions. So some other functions of the plasma membrane include, as we said, a mechanical barrier, so just separating the inside of the cell from the outside environment. That's selective permeability, so it's going to control what is allowed in and out of the cell. It's also going to help maintain an electrochemical gradient by maintaining certain ion and chemical concentrations in the fluids. Also participates in cell-to-cell communication and recognition, as well as cell signaling and receiving chemical messengers, such as hormones. Membrane lipids are composed of 75% phospholipids. So we talked about phospholipids in the chemistry chapter. So we said they have two regions. So we have a hydrophilic head or water-loving head that's going to be exposed to the intracellular and extracellular fluid. And then the phospholipids have hydrophobic tails, so they are scared of the water, so they'll turn inward facing one another, so they're kind of insulated and protected against the fluid or water environment by the hydrophilic heads. 5% composition is glycolipids, so some of those sugar lipids on the surface of the cell membrane for cellular recognition and identification, and 20% cholesterol lipids, so that's steroid lipid. It's just going to help to increase the stability of the membrane and the structure. These membrane proteins allow cell communication with the environment by allowing things in and out of the cell as needed. The proteins make up about half of the mass of the plasma membrane, and most of them have very specialized functions. Some proteins can float freely throughout the membrane and kind of just bob around, and others will be permanently tethered or attached to certain intracellular structures. Two main types of membrane proteins include integral proteins, which will span the entire length of the membrane. So things like channel proteins and proteins that allow passage of ions and molecules. And peripheral proteins, which are just kind of on the edge or the periphery, but serve more for attachment to other cells. So looking at some different membrane protein functions. So they can function in transport, so allowing different ions and substances across the membrane in or out of the cell. Receptors for signal transduction. So some membrane proteins act as receptors for certain chemical messengers like hormones. Attachment to the cytoskeleton and the extracellular matrix, just to help anchor and maintain the cell shape and stability. Enzymatic activity. So some membrane proteins act as enzymes. Remember we said enzymes were biological catalysts that help speed up the rate of chemical reactions. Intercellular joining, how we join different cells together to form tissues. And cell-to-cell recognition. So those glycoproteins, that glycocalyx, on the surface of the membrane serve as identification tags that are specifically recognized by other cells' membrane proteins. Some cells in the body are free, meaning that they're not connected or bound to any other cells. So things like blood cells and sperm cells, right, are free individual cells. But most of our cells will be bound together to form tissues and organs. So there's three different ways that cells can be bound to each other. So with tight junctions, we have almost like a stitching effect of the adjacent cell membrane. So we have a very tight connection that's going to prevent any molecules from passing through these spaces. So you find tight junctions in tissues like in the bladder or other fluid-containing organs where we don't want any of that fluid to seep through between the cells. Desmosomes are more kind of like a Velcro attachment. So the cells are still physically attached to one another, but they have a little bit more give to them. They're not anchored or stitched together so tightly. So you'll find these in tissues like muscles, where we have a lot of contraction and pulling and stretching. And then finally, gap junctions are more of a chemical or electrical cellular connection. So these first two, tight junctions and desmosomes, are just a physical connection. Gap junctions are how cells can communicate and allow ions or electrical signals to pass rapidly from one cell to another. So these are commonly found in cardiac muscle in the heart. So how do substances move across the plasma membrane? We said it was selectively permeable. So permeable just means that something can pass through something else. So selectively permeable means that some things can pass through, some things can't. So there's two ways that substances can cross this membrane. So we can cross via passive processes, which require no energy. So essentially we just roll downhill or just go, with the flow downstream. Active processes do require energy in the form of ATP. So in this case, we would be going kind of against the flow or current going uphill, right, which would require more energy. Passive membrane transport requires no energy. So just kind of left to its own devices, this would be kind of a naturally occurring phenomenon. So diffusion basically just states that solutes will move from an area of high concentration to an area of low concentration. So they're going to flow downhill, right, until it reaches an equilibrium. So there's three types of diffusion. We can have simple diffusion, a carrier or channel facilitated diffusion, and osmosis. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So the difference between these is called a concentration gradient. So again, it's always going to go from high to low, so from uphill downward. So it does not require any energy. So think if you're riding your bike down a hill, you don't have to pedal, you can just coast. The speed or rate of diffusion can be influenced by the size of the molecule as well as the temperature of the solution. So in this example, we have a beaker of water that we place a drop of food coloring or food dye into. So when you first drop the food dye into the water, right, it'll have this little ball of color or dye in the middle of the water. But after a few seconds, it will start to spread out and disperse. So the dye molecules are going to diffuse throughout the water until they're evenly dispersed and reach that equilibrium. So everything in nature and in the body wants to reach equilibrium. It wants to have that balance. So diffusion is one way that we can reach that balance and equilibrium. Generally, nonpolar hydrophobic lipid core of the plasma membrane will block diffusion of most molecules. So it's a pretty good barrier at keeping things out. There are some molecules that can passively diffuse straight through the membrane, though. So things that are lipid-soluble and nonpolar substances can pass through. So remember, the membrane is made up of lipids, right? And like dissolves like. So lipid-soluble solutes are able to dissolve through the membrane and pass through. Right? So water-soluble substances would be repelled by this lipid membrane. Some very small molecules can pass through the membrane or certain membrane channels, but larger molecules would get stuck in between the phospholipids, so they require some assistance via carrier molecules. So in simple diffusion, these lipid-soluble solutes just zip straight through the membrane, very simple. Facilitated diffusion is just where we need a little bit of assistance. So it's going to be facilitated by some membrane proteins. So certain hydrophobic molecules may be transported passively through these channels. Right? So some things that are too large to fit through the membrane itself or can't dissolve. In carrier-mediated facilitated diffusion, the substances bind to specific protein carriers. So here we have a specific protein that only will fit sugars or amino acids. Right? So only this shaped molecule can pass through this channel. So once a molecule binds to the protein, it will open, allowing it into the cell. Channel-mediated facilitated diffusion is basically just like an open channel in the membrane, so anything small enough to fit through that tunnel can enter into the cell. So there's two types of channel-mediated facilitated diffusion. So we could have leakage channels that are pretty much always open, so they're leaky, so anything that fits through can come on in. A gated channel is only going to open under certain specific chemical or electrical signals. So a neurotransmitter or hormone or some type of ion can trigger opening of these channels. So it's gated, and you have to have the key to open up the gate. Osmosis is the diffusion of solvent or water across a membrane. So when we talked about diffusion, we were talking about the solutes that were moving across the membrane. In osmosis, we're talking about the water that's moving. So it's kind of going to be in reverse of diffusion. So the water is going to be moving in the opposite direction, generally, of the solutes. Water can diffuse through the plasma membrane through simple diffusion. Since water molecules are pretty small, but it's not as quick or efficient as if they pass through their specific water channels, these aquaporins or water pores, water holes in the membrane. So the flow of water is going to occur when the water or solvent concentration is different on the two sides of a membrane. So in this example, we have a beaker full of sugar water solution. Right, so red for sugar, blue for the water. We have a semi-permeable membrane in the middle. The more concentrated sugar solution is over here on the left side. Right, on the right side of the beaker is more dilute, so it's more watered down sugar water. So via osmosis, the water from over here will diffuse to more concentrated side to try to dilute out the sugar solution and make both sides equal in their ratio or concentration of the solutes and water. So when we talk about osmosis, you may hear the word osmolarity. So osmolarity is basically just the measure of concentration of solute particles in a solution. So how concentrated is this solution? So a high osmolarity or a hyperosmotic solution would be more concentrated. It would have less water in it and more solutes. A hypoosmotic or a low osmolarity would be a lower solute concentration with more water, a more dilute solution. So water concentration varies with the number of the solute particles because they're going to displace water molecules. So we only have a finite volume. So if the solute particles outnumber the water, it would be more concentrated. So its osmolarity is going to increase. So water moves by osmosis from the areas with the lower solute concentration, high water, to areas of high solute concentration and low water. So if you think about it in terms of diffusion, it is still going from high to low concentration, but a high water to low water concentration. Whereas in diffusion, the solutes would diffuse going this way because they're high concentration of solutes to low concentration of solutes. But osmosis, we're going from high concentration of water to a low concentration of water. When solutions of different osmolarity or different concentrations are separated by the membrane, both solutes and water can cross the membrane until it reaches that equilibrium. So again, equilibrium is when you have the same concentration on both sides. So everything is nice and balanced. Right? So here we have a more concentrated solution. Right? So here we have high water concentration and low water concentration. So now some of this water is going to move over here, and some of these solutes would move over here. Living cells have limits, though, to how much water they can hold. Right? So water can also leave a cell and cause it to shrink, and too much water could cause it to burst. So any change in the normal cell volume can disrupt its function, right? Especially in neurons. Going back to structure always reflects the function. Tonicity is the ability of a solution to change the shape of a cell by altering its water volume. So different tonic solutions or different concentrations, we can drive osmosis in or out of the cells. An isotonic solution is where the solution has the same concentration inside and outside the cell. So essentially, there's no net water movement. It's already at equilibrium. It's already balanced. So cells will retain their normal size and shape. In a hypertonic solution, so hyper means kind of above the average. So we have a concentrated solution. Right? So there's more solutes in the solution than is what in the cell. Right? So there is a higher water concentration in the cell and a lower water concentration outside because it's hypertonic. So this is going to cause the water via osmosis to diffuse out of the cell to try to dilute this real concentrated solution that it's in. But ultimately, this would cause the cell to shrink or crenate and die. Hypotonic solutions, hypo means below normal. Hypotonic solutions have lower concentrations of solutes than inside the cell. So inside the cell is more concentrated. So the water, higher concentration of water outside the cell will move inside the cell trying to dilute this concentration of solutes, causing the cell to swell and potentially burst. So hypotonic causes the cell to take on water and swell. Isotonic, you have a happy cell. Hypertonic, cell's going to lose water and shrink. Water is always going to move toward the hypertonic areas. So whatever has a higher concentration of solutes, the water will go in that direction to try to dilute that solution. Active transport processes require ATP energy because we're going against gradient. So we're going uphill or upstream against that natural flow from diffusion. Sometimes solutes are too large for the channels and would require some extra assistance from ATP as well. Some solutes are just not lipid soluble, so water-based solutes would be repelled by the phospholipids, so they would also need some energy to kind of overcome that repulsion. We can use ATP energy to move solutes down the gradient if they're not able to move on their own. So some types of active transport include primary and secondary active transport and vesicular transport. So there are two types of active transport. In primary active transport, the energy comes directly from ATP hydrolysis or the breakdown of ATP. So remember, ATP provides energy when we cleave off or cut off that third phosphate group. When we break that chemical bond, it releases the energy needed for the cell to carry out its function. So using ATP, this protein can pump sodium ions across the membrane against their gradient. Secondary active transport is going to require energy that's obtained indirectly from the ionic gradients that were created initially by the primary active transport. So it's a form of active transport, but only in the fact that it's dependent and kind of a consequence of the primary active transport. So out of all of this sodium that we pumped out with our primary active transport, now we can diffuse glucose, carry glucose back into the cell through a symporter protein. So the glucose and the sodium travel together. The sodium helps bring the glucose and carry the glucose into the cell. Vesicular transport is endocytosis and exocytosis. So we're bringing things inside the cell or we're expelling things outside of the cell. So endocytosis, inside, and then exocytosis, outside. This involves the transport of large particles or molecules and fluids across the membrane and throughout the cell in little sacs, membranous sacs called vesicles. So these are essentially little sacs of plasma membrane and cytoplasm fluid. So because they're composed of the same type of material as the cell membrane, when they reach the membrane, they can just kind of fuse and bud out or bud in. But this process requires energy, usually in the form of ATP. So there's three types of endocytosis depending on what we're trying to bring into the cell. So in phagocytosis, this is kind of like cell eating. So like an immune system cell will eat a bacteria. So it will extend portions of the plasma membrane out and kind of engulf and wrap around the large particle, bringing it inside the cell into a vacuole or a vesicle. Penocytosis is kind of like cell drinking. So instead of reaching out and grabbing, it's just going to draw inward and suck in some of the fluid from the extracellular fluid. So we're not really grabbing large particles, we're taking in fluid and maybe some ions or chemicals. Receptor-mediated endocytosis is just a very specific form of endocytosis. So we won't undergo the process of endocytosis until specific molecules have filled our receptors. Right? So we won't start the process until there are 3 of the star molecules bonded. So just one is not going to trigger the endocytosis.